The objectives for this part of the lesson or lecture are: number one, define the membranous organelles and their functions, define the nucleus and its functions, explain the life cycle of the cell and its importance, go over the steps of mitosis and their importance, and cancer, how it develops and its causes. There are 5 membranous organelles: the endoplasmic reticulum, the Golgi apparatus, the lysosomes, the peroxisomes, and the mitochondria. The endoplasmic reticulum is basically the highway of the cell. Endo means within, plasm means cytoplasm, reticulum means network. So the network within the cytoplasm. The cisternae are the storage chambers within the membranes. There are 4 basic functions of the ER or endoplasmic reticulum: synthesis of proteins, carbohydrates, and lipids, storage of the synthesized molecules and materials, transport of the materials within the ER, and detoxification of drugs or toxins. The endoplasmic reticulum can be divided into 2 parts: the smooth endoplasmic reticulum, or the SER, and you can identify that because it has no ribosomes. And the main importance of that is that it makes or synthesizes lipids and carbohydrates. So it produces the phospholipids and cholesterol for the cell membranes. It produces the steroid hormones for the reproductive system. It produces the glycerides that are stored in the liver and the fat cells, and the glycogen, which is stored sugar in the muscles. The rough ER, or rough endoplasmic reticulum, or RER, has a surface that is covered with ribosomes, and it's active in protein and glycoprotein synthesis. Here's an image showing you the ER and how it surrounds the nucleus and where it's located in the cell. Make sure you know that for your test. The Golgi apparatus is the next membrane-bound organelle, and its major function is to modify and package secretions. These include hormones or enzymes, and they are released through exocytosis. Secondly, it renews or modifies the cell membrane. And finally, it packages special enzymes within vesicles for use within the cytoplasm. This is an image of the Golgi apparatus and where it's located in the cell. Next are the lysosomes. They're powerful enzyme-containing vesicles. The primary lysosome is formed by the Golgi apparatus and inactive enzymes. Secondary lysosome is a lysosome fused with damaged organelle. Digestive enzymes are activated, toxic chemicals are isolated, and basically it helps kill disease and repair the cell. Next are the peroxisomes. They're enzyme-containing vesicles. They break down fatty acids and organic compounds. However, they produce hydrogen peroxide, and they do replicate by cell division. Finally, we have the mitochondria, the powerhouse of the cell. They have the smooth outer membrane and inner membrane with numerous folds called cristae. And the mitochondrion takes chemical energy from the food. So the mitochondrion uses glucose to make ATP. This produces the energy molecule ATP, and it gives off 95% of all the required energy for the body through aerobic metabolism. The mitochondria energy production is called aerobic metabolism or cell respiration. And the chemical equation is glucose plus oxygen plus ADP equals carbon dioxide plus water equals ATP. The last membrane-bound organelle is the nucleus. It's the largest organelle and it's the cell's control center. The nuclear envelope surrounds the nucleus and separates the nucleus from the cytosol. And the nuclear pores are openings that allow things in and out of the nucleus, and they're also communication passages. The contents of the nucleus include DNA. All the information to build and run an organism is found there. And it's in a fluid called a nucleoplasm. Nucleoplasm is a fluid containing ions, enzymes, nucleotides, and some RNA. chromosomes contain the DNA information. Information is stored in the nucleus in DNA. DNA is the instruction for every protein in the body. A gene has the DNA instructions for one protein. The genetic code is a method of information storage inside the DNA strands. The nucleus allows us to study the inherited traits from generation to generation. Now, next is the life cycle of the cell. Most of a cell's life is spent in a non-dividing state called interphase. Soma or somatic cells divide in 3 stages. They have DNA replication, so the DNA is duplicated, and that means there are 2 copies of the genetic material. Mitosis divides the genetic material equally. Cytokinesis divides the cytoplasm and organelles into 2 daughter cells. Interphase is the non-dividing period. The G0 phase is where the cell carries out its specialized functions only. In the G1 phase, cell growth begins, organelle duplication begins, and protein synthesis begins. In the S phase, that's the stage where the DNA replication takes place. And then the G2 phase finishes protein synthesis and central replication. This illustration shows you interphase and mitosis. We start with G0, we move to G1, then we move to S, then we go to G2, and then mitosis begins. Prophase, metaphase, anaphase, telophase, and then cytokinesis. Mitosis divides duplicated DNA into 2 sets of chromosomes. DNA coils tightly into chromatids. The chromatids connect at the centromere, and the protein complex around the centromere is called a kinetochore. There are 4 phases in mitosis: prophase, metaphase, anaphase, telophase, and then cytokinesis. We're going to begin to look at them now. In mitosis, prophase is the first phase. In prophase, the nucleoli disappear, the central pairs move to the cell poles, and the nuclear envelope disappears. Second is metaphase, and that's where the chromosomes align in a central plane called the metaphasic plate. Here's early prophase, late prophase, and then metaphase, and you see the chromosomes lined on the metaphase plate. Next is anaphase. The microtubules pull the chromosomes apart and the daughter chromosomes group near the centrioles. The last and final phase is telophase, and that's where the nuclear membrane reforms, the chromosomes uncoil, the nucleoli reappear, and the cell has 2 complete nuclei. This shows you anaphase with the daughter chromosomes pulling apart, moving towards the centrioles, and then telophase where the nucleus is reformed and you have 2 complete nuclei. And then the last stage is cytokinesis, where the cytoplasm and the organelles split in half, having 2 complete cells. That leads us to cytokinesis. That's the division of the cytoplasm, and a cleavage furrow around the metaphase plate. Membranes close, producing daughter cells. Now, what is cancer? Cancer develops in steps: abnormal cell, primary tumor, metastasis, and secondary tumor. A tumor, or neoplasm, is in a large mass of cells, and it usually happens due to abnormal cell growth and division. A benign tumor is contained and not life-threatening unless it's large. A malignant tumor, on the other hand, spreads into the surrounding tissues. There's an invasion and starts new tumors metastasis. And this shows you an illustration of the tumor growth. You can see the group of cells, and now it's where it's starting to move and invade. In this case, it's getting into the circulatory system. When it reaches the circulation, It will move more rapidly throughout the body and has greater chance of metastasizing and going to different areas.